Brian Ogilvie is our Vice President of our consulting services, and we have a nice booklet on what our consulting team does. They do a lot of gap analysis, the due diligence review for in-licensing assets, technical writing, and he does a lot of preparation for advisory board meetings. So today Brian's going to talk on the in vitro aspects of the ICH M12 harmonization document from an industry and technology perspective. So great to have you with us, Brian, and
thanks, Maria.
Thank you for your talk.
So this is a presentation that really focuses on the in vitro part of the ICH guidance. So I don't know if you've read through the ICH guidance. It combines the clinical and the in vitro, and I'm just going to focus on that part. But basically, the overview of the landscape is really the first part of it, and then the summary of really the major points. And then I'll go through a little bit on timing. Timing is not as clear-cut as people would like, and if you look at and by the way, can everyone hear me in the back? Okay. And if you look at a lot of the comments that have been made, especially to the EMA, I think FDA got 2 comments, EMA got pages and pages of comments. A lot of them were the same about the timing isn't clear, and I would agree with that. And I think they leave it purposely ambiguous, so I'll talk a little bit about that. And then I have really a lot of comparison tables, and these come from back probably 2017 when FDA issued their new draft guidance, and it really changed a lot of things. And then they ironically issued their final guidance in 2020, just a couple years before the ICH came out, and now will supplant that final guidance. First time FDA had a final DDI guidance. So those tables are really just kind of a rehash of what we've done before, but I found that they were very popular. In fact, Andrew Parkinson and I actually came up with those, and we put together a poster at one point that turned out to be quite popular. I had someone tell me that they actually blew up the poster, like poster size, and put it on their wall to have as a reference. So because of that, I've kept those in, and then kind of covered the DDI assays of the metabolites very briefly, and then go on to the experimental conditions, considerations, really. So the appendices have a lot of detail in terms of, how things are done, which I think is important because, I can tell you in consulting, I see a lot of other, not necessarily just CROs, a lot of other reports, and sometimes the conclusions don't actually match the results, and sometimes the results and the conclusions match, but the methods are made in such a way that really undermined the results themselves. So, and then briefly cover new modalities. So there is some coverage of peptides, proteins, some monoclonal antibodies, and also some of the other larger molecules. And then conclusions. So starting off, regulatory landscape. Like I mentioned, the FDA finally finalized the 2020 guidance, which will last about 4 years. Because next year it'll be defunct. And I've heard that from 2 people at FDA, that they do absolutely intend to adopt the ICH when it becomes finalized, probably in March or April of next year. And then the EMA, of course, finalized in 2013, and I've heard EMA say several times they believe in finalizing things, and so they left it final. They haven't updated it really since then. They have updated their thinking a couple times, and I've heard straight from EMA, this is what we think. They draw a line in the sand, like for instance, peptides. They will say, we look at peptides as small molecule drugs, and so therefore we think you should put them through all the transporter, inhibition, and so on studies that you would for a small molecule. Now that can always be negotiated, but that's something I've heard before. And then PMDA, that was 2018, that was a little bit more conservative. That actually took quite a while to translate. I think we had to pay someone to translate that for us. Before they finally put out their English version of that. So I look at the ICH as not one guidance to rule them all, it's going to guide them all. In fact, like I mentioned, FDA has definitely said they will adopt the ICH guidance. EMA, I've heard directly that they will also adopt the ICH guidance in the end. But then one of my colleagues was speaking with someone last year from EMA who said, yes, but we still reserve the right to, when we're more conservative, ask for that instead. So we'll see what happens there. I thought that was an interesting comment, though. So, the ICH, like I said, usually replace the regional documents. And I don't have to read all these quotes. It's basically just the FDA saying that they intend to adopt it. And again, it won't be finalized until April 2024. So bottom line, though, I think that if you would review your current in vitro data with the draft ICH in mind now, would it change the interpretation? So in other words, are some of the cutoff values different? Are there certain considerations in the experimental design that might make a different interpretation in the end? And those are the things you need to kind of hone in on now and think about, do we need to adapt this? Is it going to change next year? Like if you have an IND that's in preparation or going for first in human studies elsewhere, is there something that might change? So just to summarize some of the major points, again, focusing on the in vitro, the title again is simply drug-drug interactions because they include both the in vitro and the clinical. It's not like the FDA where they had two separate guidances. I think for a while they had one. They went to two separate ones at one point. And covers both in one. So it incorporates many aspects that are already in the FDA, EMA, and PMDA guidance documents. There's not a whole lot here that's new. It's really just about taking not necessarily the best of all of them, but putting aspects in them that they could agree on. This was started with a concept paper in 2018 or 2019, I believe, and then working groups over that period of time until this was finally issued last May. So it took a while for this process to evolve. And then, includes mentions of transporter induction if SIPs are induced. So this is one thing that may be slightly new. I think EMA possibly referred to that in the past. And then there's much more detailed assay methods. So they actually go into, I'm actually curious how many people, by show of hands, have actually read through the whole ICH guidance document. Okay, so a few, yeah. So if you go into the appendices, there's just page after page of detail on how you should do things. And they never really put it in black and white. They always leave a little room so that you can change what you're going to do for certain circumstances. And it includes the therapeutic proteins, antibody drug conjugates, and pharmacogenetics. So the ADCs, that's something that we've actually been working on for well over 15 years. Here and there, we just have companies that will say, we think we should go ahead and put these through these studies, or maybe FDA or EMA actually ask for them. Now, ICH actually puts that into the guidance document and says what you should do with those. But as far as the pharmacogenetics, it's really just kind of touched on. And I think that there's the potential for more guidance on that in the future. I know the FDA has recently finalized their therapeutic protein guidance document. Honestly, I don't, no disparagement here, I'm not sure they knew what the word guidance meant when they put that peptide document out, because I didn't see a lot of specifics that would have changed my thinking on how to perform those studies. And then other major points that will be covered in later slides. So I'm not going to cover everything here. So timing of studies. So, for FDA, you really have to kind of work backwards from the clinical document. And what they say is, when do you need those in vitro DDI data? And the answer, of course, is before you administer that to patients. And that can be a number of settings. If you're going into oncology or something else where you have to enroll patients, then you would probably need to get a lot of the in vitro DDI data up front. And that doesn't mean that you can't go ahead with those first-in-human studies in patients, but it means that you might possibly have to exclude patients, especially in oncology where they're going to be on multiple therapies anyway. You don't want to have a problem with recruitment, and so you're going to have to get those data quite early. And I remember hearing this for the first time probably 2017, before FDA even issued their guidance document. Had a client that were coming to us in a panic, basically saying, look, we've got this first-in-human study going on in oncology, and now FDA is suddenly saying, we want you to revise your protocol to put all these exclusions in place because you don't have any in vitro transporter inhibition data. And so they were going to exclude patients or make it difficult to recruit. So that's the point of having that emphasis on patients. And then you have to collect, obviously, enough DDI information to prevent patients being excluded and increasing risk for the studies themselves. Now, if you move on to the ICH, in this case, they're saying this is a few things that were commented on, actually. So if the drug is a substrate of metabolic enzymes, you should generally have data obtained before starting phase 1. No mention of patients. So that basically implies that you would need to run some reaction phenotyping studies before you even go into first in human studies in healthy volunteers. And most people don't necessarily do that. Some people do. And so that was commented on multiple times in the EMA document. And I think that the point really is you want to know early on which enzymes are metabolizing it, if any, so that you can potentially then go forward in your once you get to phase 2 and really know what you're dealing with. But again, that's a little bit ambiguous. I would not be surprised if this changes by the final guidance next year. And then the results of mass balance studies, they say generally should be available before phase 3. I think that's probably why most people are doing. They're deferring mass balance studies until later on. But they have other parts in the guidance document where they refer to knowing information that would only come from mass balance studies before phase 3. So it's a little bit ambiguous, and that was also raised in some of the comments. Actually, I will say on the comments themselves, there were quite a few insightful comments. I think Sotara in particular had a lot of interesting comments there. A couple other pharma companies that had some good insight in their comments. There was even one that was right off the bat. The very first one was, we think that the use of the phrase victim and perpetrator is mildly offensive. I kind of laughed at that one. I always thought it was awkward. I didn't think of drugs as being victims and perpetrators, right? But I don't know if I call it offensive. And then they go on saying that, if a drug has limited absorption and so on, the relevant transporters should be identified before initiating clinical studies in patients. So again, they're going back and forth between, is this in healthy volunteers or is it patients? They do say patients here. So bottom line from this is the timing. It looks like ICH wants you to get some reaction phenotyping data early on. And that could be as simple as just running a panel of recombinant CYP enzymes. If you know from the structure that maybe it's going to be conjugated, you might have to throw UGTs in there. But I don't think from this guidance that you would have to do a full-blown reaction phenotyping study before you go first in human study. And then they talk about perpetrator potential on major CYPs, and transporters should be, again, available before administering to patients. So again, they cover that. DDI potential of metabolites. So this is a little bit interesting nuance here. With significant plasma exposure. So they define later on what significant is. It's a little bit better than what FDA had, or pharmacological activity should be considered similarly as for the parent drug, and they can generally be deferred until later in development. The problem with that is that if you have a drug that by phase 1, for instance, has 1 or 2 major metabolites by their definition, now you've got to go back later on and do those studies again. So some of our clients actually will have, in fact, we had one client had this unfortunate situation where they had as many as 12 major metabolites by definition. And what we did is we grouped them by whether or not there are acids, chain shortened, and I think there was one other category, and we put them in a cocktail. And then if they were clean for inhibition, for induction, then we knew that those were good and we didn't have to do any more. If you have a hit in that case, it's kind of a risk, because now you have to go back and look at the 3 or 4 metabolites that are in that mix and figure out which one's the culprit, or maybe more than one's the culprit. So I think there's some adjustment that can be done on that aspect, depending on the number of major metabolites you have. And I'll cover what constitutes major in a little bit.
Can I ask you a question?
Yeah, absolutely.
I mean, the first one with the substrate metabolic enzyme before starting phase 1, they are not on other medications. Healthy volunteers. What is the thought? What are they afraid of?
Yeah, that's what a lot of the comments ask too. what are they afraid of? I don't. I think it's just, that's a good starting point. I think they thought, if you can get some safety data, like what's metabolizing it, and then corroborate it in your not in mass balance, but in your first in human study. So, if you're collecting urine, if you're collecting plasma, then you can send those samples off for some supportive studies. Yeah.
Just a comment, potentially pharmacogenetics. So if you have a poor metabolizer, then you might need to exclude those, depending on this. So just say that is a risk, but that is not only in patient, but also in healthy volunteers. Would be the
Rare case.
2d6, then maybe. Yeah, well, then you might have to make a separate board or something. So yes.
But I think it's a good idea to know beforehand that it's
maybe a little too conservative. Although I will say, Roche has published this several years ago, maybe 2015, 2017. They had a drug that was solely metabolized by UGT2B10. It forms an N-glucuronide, and they ran the first in-human studies in the U.S. in the South, and they had 2 patients out of a cohort of like 10 or 12 that had AUCs that were something like 140-fold higher. And a lot of investigation showed that because I ran the panel of UGTs, they didn't see anything really except maybe 1 or 2. Then they ran an extended panel that included UGT2B10, and that was the enzyme that metabolized it. And there was only 1. And it's found now that there's a polymorphism in African American populations in the United States that are poor metabolizers for that enzyme. So that's why those 2 patients, they got lucky in a way because they detected that early on. Otherwise, they could have gotten into phase 2 and they would have had problems. I think that drug was then shelved. Yeah. Okay, so these tables really, I'm just going to highlight the differences. And some of the highlights. So for instance, ICH, now they've spelled out the UGTs. So previously, the other guidance document just mentioned UGTs. So FDA even just says UGTs. PMDA calls out UGT 1A1 and 2B7. And I believe that EMA mentioned something as well. So now they spell out which ones to add. But then the ICH also notes that if the drug is not found to undergo significant metabolism by CYPs, then other enzymes should be investigated. And obviously that makes sense. In fact, one of the first things I have always asked is, can I see the structure of the compound? If it's got a nice phenolic group on it that looks like it's going to be conjugated immediately, then maybe human liver microsomes and NADPH isn't the best system to go into. Or just looking at a recombinant CYP enzymes, you should probably add something like at least the UGTs. Sulfotransferases can also be added. So that's the main difference. I'm not going to go through these as quickly as I have in the past because we do have a little bit of time. So again, everything in red, ICH, and whereas FDA notes this is for transporter substrate. So FDA says yes if hepatic metabolism or secretion is greater than 25%. ICH also notes if the target is in the liver. And I think this is important if you consider drugs like metformin, where the target is in the liver, and it's also taken up by OCT1. In fact, there are other drugs like this. The target is not necessarily in the liver, but some of the triptans, for instance, there'll be substrates for OCT1, and that's a rate-determining step in this metabolism. Even though they're metabolized by MAOs, there are polymorphisms in OCT1. So knowing whether or not it's a substrate early on can be important. But they do call out if the target's in the liver, probably referring to metformin. And then they also talk about for renal secretion of greater than 25%, that's the same. But they also call out that if renal toxicity is observed. And so that's something that you would need to investigate early on as a victim of transporter as a substrate. And then for the ICH, they've now added, and this is actually something that was is mentioned in brief in the PMDA. So MRP2, OCT1, OATP2B1. And I think the scientific literature has really become mature, especially on OATP2B1 and OCT1. But they leave it ambiguous. Additional transporters can be decided on a case-by-case basis. And so this really puts the onus on the sponsor to decide what those case-by-case bases are. I think the last thing you want to do is wait for the regulatory agencies to tell you what you need to do. So you need to look at the structure, find out if other transporters are involved first, and then maybe add some of these if that's important. And then, finally, EMA had the other transporters can be investigated, but it's by and large the same across the different agencies. And then when it comes to a simplified interpretation of victim potential, so this would be when you have a net flux, like it says, greater than 2. Also, this is an interesting point. They don't change it here, but every guidance document before this always has greater than or equal to. And I'll show you that later on. The ICH has dropped the greater than or equal to and just put greater than. This is one case where they didn't, but they do point out that where you have the positive impact before had to be net efflux greater than or equal to 2 and significant inhibition by one or more known inhibitors. ICH now says significant inhibition has to be 50%. So that means when it comes to your positive controls that you're using, you have to make sure that you've validated enough so you can see 50% inhibition of a prototypical substrate. And so that holds true for all of these: hepatic uptake and renal uptake. And also from the appendices for this section, they note that passive permeability must be considered. And so what you would do in this case is you compare that compound, your test compound, to a high permeable control. And you can use saturating concentrations, or you can actually use transporter inhibitors. So to know the impact of passive permeability, either use a very high saturating concentration or put a cocktail of inhibitors in to see what the passive permeability is. And then you validate that in that system. And then they also talk about, in some of the other aspects that are in the appendices, you have to have total recovery that's greater than 80%, and so on. So there's a lot more detail, like I mentioned before, that you can find in the appendices more than what's on these tables. So when it comes to drug metabolizing enzyme inhibition or perpetrator scope, basically it's the same, but the ICH now adds something similar to what PMDA had in the past. They don't spell it out in their table, but they put UGT 1A1 and 2B7 and others. EMA only call that 1A1 and 2B7. But now they're saying if you have direct glucuronidation, then it's saying look at UGTs including 1A1 and 2B7. So basically, if you know you have direct glucuronidation of your drug, then you need to look at it as an inhibitor of UGTs as well. So it's not just the CYP enzymes. ICH also, they have this call out here at the bottom that when an investigational drug is to be used with another drug that's mainly metabolized by direct glucuronidation, it's important to evaluate that in vitro to see what the effect is. So that would be a case where you have, it could even be a fixed dose combination. If you know that your fixed dose combination is one of the other drugs in the combination is metabolized by UGTs, you would need to look at the other one for metabolism or inhibition of UGTs. That could also be a case where you have a population that is always being administered a drug that's continually conjugated. That would be a case where you need to look at UGT inhibition. But again, it's not always clear. They leave latitude. They leave some interpretation there. And I think that from the comments, I'm hoping that some of these things will be tidied up in the final guidance, but we have to wait till next May for that probably. So these are the interpretation slides. So this is when you have hepatic CYP inhibition. And one thing to note is that ICH talks about what they call no effect boundaries. So they say that if the Ki unbound is greater than, what do they call it, 50 times the Cmax, don't worry about it. So you can also turn that into an equation that basically comes out to Cmax unbound over Ki unbound. If that's greater than 0.02, you've met that no effect boundary and you're good. You don't have a perpetrator potential. So that's equivalent to what EMA has. So these are still pretty conservative for the most part, but they are all equivalent. And if you look at the reasons for that in the equations, and I heard a funny joke at the Marbach this week. Someone said, just a black box warning for my presentation. It does have equations in it. So if you these aren't complicated equations, right? I mean, even I can do these. But I just want to point out some of the differences. So FDA and PMDA have this 1+ factor, which is why you get this greater than 1.2. And you'll see later on it might be greater than 10 or greater than 11. And that's the only reason for that difference. It's just missing those 1+ factors. Both the ICH and FDA now cite our paper from 2015 where we had to prove to people that you can actually extrapolate Ki from IC50 values. So now they allow you to do that. You can just take the IC50 value, and if you've run it at Km, you can just use that Chang-Prusoff equation from back in the 70s and extrapolate what the Ki is. Interestingly, though, when it comes to transporters inhibition, and I'll show you that in a little bit, now you're doing transporter inhibition studies at substrates much, much lower than Km, where Ki would necessarily equal IC50 anyway, and there you have to use Ki. So there's a little bit of a discrepancy there. I think that's kind of interesting to me. So going on then to intestinal inhibition. So now we've got, again, the same no effect boundaries. ICH talks about the dose in 250 mils, and basically the way it's written, it's talking about the Ki here. They don't put the unbound term here for the intestinal, and that comes out to greater than 10. Again, I'm just highlighting it's greater than as opposed to greater than equal for some reason. And because it's missing that 1+ factor, it's 10, not 11 like it is for FDA and PMDA. So you can also write that equation as simply Ki is greater than 0.1 times the maximum clinical dose in 250 mils. And so they're keeping the dose in 250 mils. That's been pretty consistent across these guidance documents. So now for irreversible inhibition, this is a couple of interesting changes here. So for one thing, some literature came out between the time the FDA finalized their guidance in 2020, where they have once you get Ki and K in ACT, they put a 50-fold, and so to PMDA, 50-fold safety factor in there. The literature came out and showed that grossly overestimates it, and you're going to almost always end up with a false positive. And so ICH has now adopted this 5-fold factor here. So that's the good news. There's a new equation for that, only because it has the 5 value. And again, it's greater than 1.25 for your cutoff value. ICH also has some additional experimental conditions in the appendix. And so that factor has been recalculated. And the PMDA and EMA also recommend estimating unbound inhibitor concentration in vitro, even though they don't put that in the equations. So whereas you've got Ki unbound here, it's not listed here in the equation, but it is listed in the text. So I just want to point that out. So if you're looking at this comparison of equations, just know that the Ki unbound should be used even for EMA and PMDA, which hopefully we won't have to worry about next year. So for interpretation of irreversible inhibition of intestinal CYP3A, so this is a little bit of a mystery for me because until I heard people talk at the Amsterdam meeting, the DMDG from EMA, kind of explain this. But this was a mystery from 2020 because suddenly there was no R2 gut equation. It just wasn't there. PMDA left it. And so at that time, I was actually advising clients that, if you want to know what's happening when you have CYP3A4 inactivation in the gut, just use the PMDA equation and see what the effect is. And anyway, what it turns out is that, again, literature showed that this grossly overestimated inactivation in the gut. And what actually happened was that if you go back to this one, this equation covered it just fine. And so ICH took it out, and they explained that verbally at this meeting in Amsterdam. So that was good intelligence to know that, because they're not explaining the rationale in these guidance documents for the most part. So moving on to induction. So these, again, are pretty much the same. The one thing, these are basically just taking the R equation that's similar for the R3, FDA, PMDA, and even EMA. They have this R3 type equation there. And again, the cutoff is exactly the same. But the ICH actually says sponsors are encouraged to measure concentrations of the parent drug in the medium on the last day of incubation. So this is like the spent media analysis that's referred to in the FDA. And they say that with hepatocytes and protein binding should also be considered. And then when measured concentrations are substantially lower, sponsors should discuss the potential impact. They don't tell you what to do. They said just discuss it. So I think that's good in some ways, because if they said, hey, if you have nothing there and it's just a property of your compound, what are you going to do? Are you going to run induction studies at 4 hours? Are you going to run? In other words, you have to be able to explain it. Explain maybe why you can't get it. Are there other systems? Can you use a reporter gene construct, something else that would satisfy them, that you have a good true positive, or true negative rather, in this case. So for transporter inhibition scope, so this really hasn't changed a whole lot, but ICH does say that same as FDA and PMDA. Ooh, we got some music now. But they do add BSEP, MRP2, OCT1, and OATP2B1 on a case-by-case basis. And these are kind of, me personally, I would almost add OAT2 and OAT7 because I think those are kind of also showing some data in more recent examples. But certainly OATP2B1 and maybe even 1A2, the data are there. And I think that on a case-by-case basis means that you need to know your compound as a sponsor and you need to be able to justify why or why not you're including those additional ones. And then when it talks about time-dependent inhibition of OATPs, as FDA did in 2020, because that was well known, I think there was some literature published that shows that this also occurs for several other transporters. And so the time-dependent inhibition, the mechanism, I think, I can't explain it directly, but I think it has been described. And so they're saying you should follow the literature. And so ICH leaves it open to, hey, this is going to be a guidance document that's static, but science evolves, look at what's happening, and go on from there. So for the in vivo assessment, this is interesting. So if data are used for PBPK, determine KI, I mentioned that they wouldn't let you use the IC50 as they would for before, so I'm not really sure why they just want a more definitive value for the KI there. But then the drug concentration can't exceed solubility to cytotoxicity, so they're acknowledging that you can't change physics of the compound. That's just, it is what it is. And if high enough, but this is the kicker, if high enough concentrations are not reached, in vivo assessment is recommended. So what does that mean? I mean, does that mean you go in, in an animal model, is that going to tell you what happens for in vivo induction in humans? I don't think that's necessarily relevant. Does that mean you have to go to a clinical study and look for a signal there? There's a lot more being made of biomarkers and other things that you could look at potentially, but it doesn't explain that. So I think clarification is needed there. I think there was one comment on that. I don't think people picked up on this. It was just kind of this throwaway statement that in vivo assessment is needed. And then also from the appendices, they do mention, this is very specific, but that vesicles can actually be used for mates. And then there's other details that are present in there in terms of experimental conditions. And really that would kind of be part 2 of this presentation. I could probably go through the whole appendices themselves and go through great detail what's required there. So intestinal PGP and BCRP, the equations and cutoffs, these again are pretty much the same. Again, just rearranging this, the way they put it in the ICH comes out to essentially the same equation. All these are the same. ICH does note that, and this is a hot area of investigation right now, that other cutoff values can be proposed if justified based on your systems. And I know companies have successfully done this for FDA and EMA. And so you always have the option to validate something with your own cutoff value. So calibration is really a hot topic, and I think that some companies are really investing hard in that so they don't get stuck with these false positives in some cases. So when it comes to the OATPs for inhibition, again, everything is pretty much the same. The only thing is that for whatever reason, ICH does not mention the blood to plasma partition ratio. So it's covered elsewhere. And if you want to see a nice discussion of this, Andrew Parkinson actually wrote this up in 2019 because we were looking at the differences between the PMDA and FDA. And he goes into great detail about why it really is all the same. So for the OATs, OCTs, and mates, the one big change here is that everyone freaked out when FDA originally put this cutoff for mates as 0.02, and so they actually relented and put it back to 0.1. ICH said, no, we're going to put it back to 0.02. So that is more conservative now, and whether or not that stays, that's open to, we'll find out. They don't explicitly cover, and this is a comment that people made also, you have these cutoff values for these transporters, but you don't have it for BSEP, MRP2, OCT1, OATP2B1, which you said we should consider. And I think it's probably because the science isn't quite there yet. I mean, the cutoff value takes a lot of data to really decide what it's going to be. So then going on to DDI assays with metabolites, this is an interesting one. So they said generally not needed if there will be a clinical DDI study of the parent. At first, it seems perfectly logical, right? Because your metabolites will be there in the clinical system. But what, and then this was in a comment, I didn't think of this beforehand. what if your metabolite inhibits another enzyme? So you're doing a clinical midazolam study because your parent inhibits 3A4. Metabolite doesn't. Okay, so you think, okay, we're clean. But if that metabolite actually strongly inhibits another enzyme, you wouldn't have covered that in that clinical study. So that was an interesting point to me. And then when it comes to metabolites as substrates, this is a little bit more specific. But if a metabolite might have a safety impact, so if you know that a metabolite's important from either pharmacological activity or has an off-target activity, or if it's on target but it's greater than the parent, it's different than the FDA guidance in that respect. They just say greater than the parent, not more than 50% of the activity. And then they have additional details, obviously, in the appendix. Metabolites as inhibitors, so they say there's a little bit of a distinction here. So yes, if the AUC metabolite is greater than 25% of the parent, which is similar to FDA, but then also greater than 10% of total drug-related material. So again, they're talking about mass balance studies. So at which point do you need this? Or you can just have really good radio-labeled in vitro metabolite ID studies. That might be one approach to take. So a little bit more consistent with the more conservative 2013 EMA approach. FDA concentration, or consideration rather, of polarity has been removed. So that was very confusing. I don't know if anyone was familiar with the FDA, but they talked about the percentages were different if it was the metabolites were more or less polar than the parent. Similar approaches were then used for transporters, CYPs, and generally no assessment of metabolites as inducers. Now, this was one exception because the idea is that if you're doing an induction study in vitro, then the metabolites should be produced in the hepatocyte, so you should see it there. But if it's a prodrug situation, then that might not be the case, and so they're saying you might have to actually look at that, or if it's formed extrahepatically. And I have had a few cases recently, actually, where we have sort of this mystery metabolite. It's not formed in hepatocytes, not formed in intestinal microsomes. In the clinic, that metabolite is major. We don't know where it's formed at this point. Highlights the need for mass balance studies, again, saying made before phase 3. So some additional considerations from the appendices. They have more details there, list of drugs. So they have, just like FDA and EMA had this list of drugs, substrates, things that you could use as clinical index substrates and so on. Now they have that listed out for not only CYPs, but UGTs and transporters as well. And then they have additional predictive modeling approaches. So they cover the basic static mechanistic models that I've shown you in these tables. And they talk about the PBPK and how it can be used. So the applications of modeling, I think it's almost expected these days that you're going to have to do some kind of modeling at some point. I think most companies want to do the modeling. So it could support clinical recommendations when a clinical DDI study has not been performed. Decide if a clinical study needs to be done. These are things that we already know to some extent anyway. But then from the Marbot meeting, had a lot of discussion about cases where PK submissions were or were not accepted. And so Ping Zhao, and I think someone else from FDA was discussing this, they don't have really good data on when it's not accepted. They just know how many submissions there were. And you can get to some of those data by looking at the approval process, the approval package rather, once it's complete. Or if it goes to an advisory committee meeting, you can look at the briefing books from both sides and see the rationale there. But for the most part, it's really difficult to decide or to tell why a PK submission was rejected, unless you do a FOIA, which is in the United States, that's a Freedom of Information Act request, which I've never done, and that takes apparently a long time to do. So then moving on to test systems, they cover a little bit more detail. So they say human liver microsomes, a pool of 10 donors is suggested. This was funny to me because we've been doing this for using 150, 200 donors in a pool. One of the comments said, why do we have to use 10 microsomal donors? For induction, we're only using 3. I don't know, I thought that was easy. S9, it talks about it contains the other enzymes and that you can control the activity of those with cofactors, except for some of the enzymes like the ADHs and the ALDHs. And then for cytosol, again, adding cofactors is appropriate to control which enzyme systems are active recombinant human CYP and UGT enzymes. The salts weren't actually covered here, although they are available, and we certainly have run some studies for recombinant CYPs in the past. And then for hepatocytes, this is where they talk about phenotyping and inhibition experiments, that you should pool those from at least 10 donors. And so this is something that we've done again for years. We even have a product called Cryostax. This is from the legacy Xenotech, the Kansas City site. We had a patent on this. So you can actually select the donors that you're going to be putting into that pool. And then, whereas for induction, you're using 3 individual donors. Now, before anyone gets real excited about this, because it says when a single culture is fully validated, they would allow you to go ahead with an induction study with that single donor instead of using 3. But what that means is you have to go through this whole rigorous validation protocol, which they put in the appendices. And then that means when your lot of that hepatocytes runs out, you've got to start all over with that process. So probably not all that practical. It's interesting, though, that human liver microsomes are the reference system for most assays, but they do cover hepatocytes and recombinant CYPs as needed. And they also talk about they would like to have that full validation, as I mentioned, again, for the single lot, if that's used. Also for induction, this is something that we've known for a while. mRNA, we used to, back in the day, used to look at activity. And then I think Pfizer made a big push that, if you have something that's a mechanism-based inhibitor, that now you would need to look at mRNA as the endpoint. So that became the change. But now they're saying that because 2C19 mRNA is not induced as much as other enzymes, you should look at activity. So that's, again, something that we've been doing there for quite some time. Also, the test article concentration for induction is only now 15 times Cmax instead of 30. So that's a bit of good news. And I'm going to cover these pretty quickly, but this is interesting. So now, for the IC50 fold shift method, previously they allowed you to go ahead and just use that without a dilution. Now their new method is appearing to require 2 experiments. And the reason I say that is because you would first have to do IC50s for direct inhibition, so this is reversible inhibition. And then you would have to go back and do an IC50 with that 10-fold dilution. And so there were a lot of comments saying we don't agree with this. And interestingly, the goal here is obviously to decrease the amount of direct inhibition that you see in the IC50 shift. But the dilution absolutely has to be done for the KI, KNAC determinations. But we actually detailed this in a 17-page final manuscript, if you can believe that. It should have been 2 or 3 back-to-back papers, probably. This was back in 2011, and several of the commenters actually referenced that. So a little bit of vindication there that people were paying attention to it 12 years later and made it into the comments. So therapeutic protein DDIs, they say that in general the risk of DDIs is lower, but in vitro assays that are applicable are generally not applicable to proteins. So that's kind of the good news, saying you don't necessarily have to run those. They go on talking about the in vitro potential between monoclonal antibodies. That really comes down to the mechanism of potential DDI. So whether or not that could be pharmacology and clearance of the monoclonal antibody, or any co-medications in the particular patient population. I would note that there's a difference between proteins and peptides. ICH does not define what that is, and that was another comment that appeared frequently to EMA. So, and as I mentioned early on, EMA, I know, has said as much in the past that peptides can behave like small molecules. So they always point to cyclosporin because it's a well-known transporter inhibition, even though it's a cyclic peptide and so on. So pro-inflammatory cytokine-related mechanisms. So certain therapeutic proteins may exert an indirect effect, and again, we've known that for quite some time. The increase in cytokine levels as a result of drug treatment, so this is if you have an immunomodulator, can be transient or persistent, so you need to consider that increase when determining whether to conduct a DDI study. They have also the investigational drug is a cytokine or a cytokine modulator, so an immunomodulator, if you will. You should consider whether to perform a clinical DDI study. There are other minimal details provided in the guidance, And I would mention that, for quite some time now, we actually have a patent on 2 different methods that allow us to treat blood, collect the PBMCs, profile the cytokines that are released from immunomodulators, whether they're large or small molecules, and then look at suppression of P450 enzymes, or other enzymes for that matter. Antibody drug conjugates. So this is good for me because we've been doing this approach for years. It's saying basically that you have to take the small molecule part of it, even though most of it are pretty well known. I think if you're using an ADC where you have the payload is well characterized, you could probably rely on previous data. Everyone looked at those for inhibition. Also, the amount that's released is relatively small, so there's a very low level exposure. But they also talk about the linker, and there's some more information here about potential to induce and even inhibit transporters with the linker, and the payload should be addressed. And then it goes on a little bit further. It might be necessary to look at it as a victim drug. I think you could probably argue your way out of this if you say that the concentration of the payload and the linker is so low that any metabolism, really wouldn't matter. I haven't put that to the test yet, but I've certainly made paper arguments similar to this in the past that at least FDA will listen to. And like I mentioned, we've been looking at these kinds of studies for years now. So conclusions, harmonization is obviously the goal. They've been meeting for years trying to compromise. And when you have compromises across different agencies, no one's going to be happy with everything. I think that's the idea. They're still hashing it out. And there were obviously a lot of people in the industry that weren't necessarily happy with some of these things. I do think I'll put it this way. I'll be surprised if they don't consider some of these comments. And if they just leave things the same without acknowledging those comments, that would be a surprise to me. It incorporates some details from EMA, PMDA, as I mentioned, much more detail in the appendices, and then additional modalities that are included. So my bottom line really would be to consider adopting some of these strategies now if your IND won't be submitted until 2024. And I think that, when you're in the business of drug development, you always have to consider that science is evolving, and the data that we're getting tomorrow might be different than what we're doing right now. And that's just to be expected. So the guidance does leave that door open for those things to change. So just because it's in guidance doesn't mean that you absolutely have to do it that way. Some people that you might meet at certain regulatory agencies might feel a little differently than that. But I think for the most part, you can do what makes scientific sense and move forward with that. But consider the ICH guidance now, even though it won't be finalized until later. So that's what I've got for you today on that. Just focused on the in vitro parts, and I'll take any questions. So thank you.